So Dr. Shori Brightman is a certified genetic counselor at Cincinnati Children's Hospital, where she has dual roles as a genetic counselor and as a laboratory genetic counselor in the Laboratory of Genetic and Genomics. Did I say that right? Yay. Together with Dr. Brittany Simpson, they established the Epigenetic Syndromes Clinic, in which they have had the pleasure of following patients with Rubinstein-Taybi syndrome, as well as some related conditions. Diana also sees patients in the eye genetics and the general genetics clinics, and she enjoys a diverse range of patients and providing support and answers to patients and families. So this is being recorded. We'll be able to share it later. At the end, I ask that you stick around for questions, and we will give you a link to a response survey, and in that we will be able to get information from you to share a recording or Dr. Brightman's slides if we're able to do that. Okay, so we're going to go ahead and get started. I'm going to stop sharing my screen, and Kelly, is Dr. Brightman a co-host so that she can share hers?
I have the ability to share my screen.
Perfect.
All right. Does my screen look okay?
It looks great.
All
right. Do we have on the closed captions, Kelly?
Yeah, is it showing up?
I'm not seeing them, but that doesn't mean anything. Is everybody seeing closed captions? Me a thumbs up for those of you who have your cameras on. Nope, no closed captions yet. Here. I don't know why it's not working now. We had it working earlier when we practiced. All right, we'll keep working on that. You go ahead and start, Diana. Oh, it's working from Katrina.
Perfect. Okay. Thank you so much for inviting me to speak with you all. I think it's so exciting, thanks to the magic of technology and maybe after effects of COVID, that we can do these things virtually sometimes and get to make these connections from all around the world. As Celia mentioned, I worked together with Dr. Simpson to establish a clinic at Cincinnati Children's, where we were seeing many patients with Rubinstein-Taybi syndrome and some of these other related syndromes. Some of you may have heard that Dr. Simpson recently had a baby, is on maternity leave, which is really exciting. Most of these slides are ones that she and I had presented together at the Rubinstein-Taybi syndrome conference in June, but I know that not everyone was able to hear it. So some of these slides will be from that presentation. So Dr. Simpson was not able to be here today since she is on maternity leave, but very happy for her, and she is doing well, as is her baby. And let's go ahead and get started. Okay, so this talk is Nice Genes, What's Your Type? I guess I should have changed the date. Sorry about that. Okay, so we're going to start out by talking about genetics and epigenetics, which is a term you may or may not have heard before, and it could mean a lot of different things. So we'll talk more about that. We'll talk about what CREB-BP and EP300 are, what they do, and why they're important for people who have Rubinstein-Taybi syndrome. We'll also talk about testing for Rubinstein-Taybi syndrome, and then also talk about what are these different technologies that we use when we're trying to interpret one of these genetic test reports. And we'll talk about RTS type 1 and type 2 and things that are the same and different between them. And then we will have some time for questions. So let's launch into it. So first, what is genetics? So, moving this. Okay. So before we start into more about Rubinstein-Taybi syndrome, I think it's great to start off with genetics and talk about what is genetics. So genetics refers to this overall topic of genes, which can be thought of like hi, not right now, sweetie. Thank you. Okay, sorry about that. So this can be thought of as a recipe book or instruction manual that tells our body how to make our bodies and then do everything that our body needs to do every day to function. So these individual instructions or individual lines in this blueprint are genes. And they give the body these instructions for how to then make some machines that do various jobs in our bodies. So in this case, you can see we have all these little different types of machines that can do different work in our bodies. Okay, so then these blueprints that we use to make machines are in some ways complex, but in others kind of simple. And we'll come back to this when we talk about genetic testing, but our blueprints have this unique code, which is our DNA. And these blueprints are all written in 4 simple components, which are easiest to refer to as the letters of C, G, A, and T. Okay. And then we can add in the next level, which is that, and if someone's just joined, if you can mute yourself, that would be great. Okay. So we use these letters in the code to then tell us what amino acid or kind of building blocks of the machine to use. So for example, we have a sequence here of a gene. Oh, sorry. And so this sequence, each of a set of 3 of these bases gives us specific instructions for which amino acid to use. And so we can think about these then amino acid building blocks where we could put together all of these pieces, kind of like putting together a whole bunch of Legos to make one of these machines. And so the instructions to make this machine were here in the DNA code, and that gave us the instructions for which pieces to use for which of these amino acids to build together into one of these proteins, which in this case is a machine that's going to be able to do something for our bodies. So as an example, each of these 3 letters, so 3 letters at a time in the DNA code for a different amino acid. And then we're able to use that information to say which of these amino acid or Lego pieces we need. So for instance, in this case, this is coding for an amino acid called phenylalanine. And so it's telling us which of these amino acids our bodies need, and then they'll put them all together to build a machine. Okay. So this is some of the building blocks of how we can have these instructions in a gene like CREB-BP or EP300, which we'll talk more about, that can tell our bodies how to make the CREB-BP or EP300 machine that we're going to talk about more. So what is epigenetics? So just like we need some different materials to build different types of buildings, we need different proteins and different ingredients in different parts of our body. So for instance, our hearts need different things, different proteins than our bones need. Or if we need to repair a building, we might need different materials and we might need different materials and things for that building than if no repairs are needed. So it makes sense if you think about it, right? Hearts are very different from bones. We need very different things. Okay. All right. So what is epigenetics? So our DNA instructions are the same in all of these different parts of our bodies. They're the same in our hearts, they're the same in our bones. But so how do our bodies know which parts of this instruction manual to use in different parts of our bodies, or when we need these different parts of the instruction manual? And basically the answer is epigenetics. Epigenetics is one of the ways that our bodies can know which genes to turn on and which genes to turn off at different times. So the way that this works is that our bodies can mark which of these instructions or which genes need to be turned on. It's kind of like if you ever were taking notes and you use some of these little tabs, these little sticky tabs that you could put in your textbook or your notebook to know, oh, I should open up here, or this is an important section that I need to come back to. Our bodies have marks that they can put on our genes that help our bodies know this should be turned on or this should be turned off. And that ability to turn things on and off is really important, because just because we needed something at one point in time, we might not need it later. So that is really crucial that we can do that. So if we're thinking about what are these different marks, these marks are a variety of things. So before we were just talking about the sequences in the DNA. So that's within this purple wound structure here. But this DNA is actually wound around these protein cores called histones. And these little histone proteins can have modifications put on them that are associated with gene activation or repression. And in addition, the DNA itself can be methylated, which can also give our bodies information about if a gene should be turned on or off. And then the other element is if our DNA is really unwrapped and open, like this example, then it is accessible and we can turn on genes. Whereas if it's really tightly wrapped like this, it's closed, it would be hard to turn those genes on. So these are the types of epigenetic changes that we talk about in terms of regulating gene expression. So there are lots of these different types of marks, including DNA methylation, and then histone methylation and histone acetylation. So that's kind of like what these tabs are. And then certain types of marks are associated with genes being turned on and expressed. So then we can make those machines from these DNA instructions, or turned off and repressed. And then we're not going to make the protein from it. We're not going to make those little machines. So, for instance, when we were talking about different parts of our body needing different things, you could have, let me, I can hide this, maybe, or maybe not. Okay, so in terms of our hearts, there's some genes that our hearts need to be turned on to do all of the things the heart needs to do, but then and so these genes have those activating marks on it, which is part of how our bodies know to make protein from those genes. And then there are different genes that are important for bones, then we don't need those in the heart. And so those are repressed and turned off. And it's the marks that are on here that tell our bodies to repress those genes. And then you can have the opposite if we looked in those bone cells, and then we would have the heart genes are turned off, they are repressed, and then the bone genes are turned on. So in addition, it's important to know that these marks don't have to stay the same all the time. You might need some genes, for instance, in your heart that are turned on forever, but you might have some genes in your heart that you only need if there's an injury to your heart, or maybe you only need in your bone if you're healing a bone fracture that you might not need all the time. And so it's really important that our bodies are able to put on these marks and take them off. And some of these little machines that we talked about earlier are, in this case, we're using like enzymes or these little machines that can put marks on, and then there are some little machines that can take marks off. One of these important marks that we are going to talk about today is histone acetylation. All of these marks are important, but histone acetylation we're particularly focused on because of how important it is for Rubinstein-Taybi syndrome. So for histone acetylation, usually these marks are associated with genes that are activated, meaning we're able to make protein using these gene instructions. So both CREB-BP and EP300 genes code for these CREB-BP and P300 proteins that are enzymes that or machines that put on this histone acetylation mark. So these little machines put on that activation mark, and they tell our bodies that a gene should be turned on. So a little bit more about CREB-BP and EP300. So as we were saying, these are the genes that encode the machines that we'll call CREB-BP and P300. And they put on these lysine marks. So these are enzymes that are called lysine acetyltransferases. And then this is really important for turning on genes. So these are a way of putting on these activation marks. Both CREB-BP and P300 can do this. They put on these activation marks that tell these genes, open up, turn on, start making the protein. We need it now. So CREB-BP and P300 are really important. These proteins or machines help us use our DNA instruction manual and help us turn on the genes that we need when we need them. And it's not just one gene, it's many genes through all of our DNA that they help regulate. So there are many of these, and we use them throughout this whole instruction manual to put those marks on and make sure we have highlighted and marked, and we're going to be able to turn on and use the parts of our manual that we need. So in people that have Rubinstein-Taybi syndrome, they don't have as many of these working machines. And so there could be some differences in how we're able to mark those genes that we need to activate. So this change in the overall ability to put these marks on changes our ability to open this book and can result in lots of different changes in different people. And we see this is part of why we see a lot of differences in terms of different symptoms from person to person in people who have Rubinstein-Taybi syndrome. So we can also think about this in this way. So the change in this overall ability to open the book, as well as to use the book, an instruction manual or blueprint, changes the way the body uses the manual. So for example, if the body is trying to build and maintain a castle like this one, there are a lot of machines that help with that process. For people with Rubinstein-Taybi syndrome, that means that some of those machines will be the same, while some will be increased, and then others may be decreased or even absent. And then this changes the way that our body is functioning, just like that could change the way that we work on this castle. And it can change the way that our body is functioning in different ways for different people. which again is part of why we see so many differences and variability from person to person. But we are learning more over time about Rubinstein-Taybi syndrome and about some of these differences that we can see. So we can also think about this like that the body and our big instruction manual can also be thought of like a music score. And CREB-BP and EP300 are the conductor of all of these processes that are happening in their body. They're conducting this whole orchestra. And these machines have to keep the body in sync and to play the right music at the right time in the right part of the body. It's also something that also comes up sometimes is that there are other genetic conditions related to CREB-BP and EP300 other than just Rubinstein-Taybi syndrome. So this was much more recently described. So we know that there are lots of changes in CREB-BP and EP300 that can cause Rubinstein-Taybi syndrome. Sometimes it's a deletion of the whole gene or part of the gene, and then sometimes it's a different type of change in the gene. But more recently, it was discovered, and usually these are changes that cause a loss of function of a copy of CREB-BP or EP300. And more recently, it's been found that some types of changes in CREB-BP and EP300, which is shown here in red at the top, can actually cause a different condition that presents a little bit differently. This is called Menke-Hennequin syndrome. Some of you, I know we had a few attendees at the conference who had Menke-Hennequin syndrome. It's not as it's even more rare than Rubinstein-Taybi syndrome, but it is a condition that we also see, and there are some things that are similar between Menke-Hennequin syndrome and Rubinstein-Taybi syndrome, but there are also some differences. And this is not unique to CREB-BP and EP300 or Rubinstein-Taybi syndrome. There are other genetic conditions where we've started to learn more about how a different type of genetic change in a different part of the gene might cause different symptoms and a different syndrome. And some of the research on Menke-Hennequin, interestingly, has also included information about facial features. I know many of you know that with Rubinstein-Taybi syndrome, there are some characteristic facial features. And there's this software that could take pictures of people and then make a kind of an average of the facial features for different genetic conditions. So this has happened, people have worked on this for Rubinstein-Taybi syndrome, which is what you see here on the bottom, that this is kind of the average facial feature from the data that they had for Rubinstein-Taybi syndrome. And interestingly, if they invert this picture, it looks pretty similar to some of the facial features that are seen in Menke-Hennequin syndrome. So in some ways, and we'll talk more about them, but in some ways, Menke-Hennequin syndrome and Rubinstein-Taybi syndrome are kind of like the opposite. In one of them, Rubinstein-Taybi syndrome, we don't have enough of that working CREB-BP or EP300. And in Menke-Hennequin syndrome, it's like we have too much of CREB-BP or EP300. We know that having the right amount of CREB-BP or EP300 is really important, and that we have symptoms if it's either too much or not enough. So in terms of Menke-Hennequin syndrome, so far there are 3 subtypes, and they are based on the variant location. And these are different domains or regions of the protein. So there's the IDR4 domain, the ZZ domain, and the TAS2 domain. And so Menke-Hennequin syndrome like Rubinstein-Taybi syndrome, is caused by a difference in the way that these machines, CREB-BP and EP300, are regulating the body's functions. But it does something different other than just decrease the amount of machines. So it might be that there's too much, or that the machines are doing different things or working in a different way. And we're still trying to learn about exactly what they do. But we think that most likely it's that it increases the amount of the machine, but I think more to come with more research. So we really do think that that balance of the right amount of CREB-BP and EP300 is really important. Some of the common characteristics that we see in individuals with Menke-Hennekam syndrome are intellectual disability, autism, some autism-like behavior, recurrent infections, feeding difficulties, short stature, small head size, and then only some of them with birth defects. We also know that some people with Menke-Hennekam syndrome have been known to have some vision problems too, but not all. And we're still learning about Menke-Hennekam syndrome. I know you all know that Rubinstein-Taybi syndrome is rare and Menke-Hennekam syndrome is even more rare. So let's transition now to talking about genetic testing for Rubinstein-Taybi syndrome. So how do we test for Rubinstein-Taybi syndrome? So there are multiple ways, and depending on, for each individual or family, and depending on when they had testing, they might have had different types of tests done, if they had testing done at all. At one point in time, we didn't have genetic testing for Rubinstein-Taybi syndrome. It was based on clinical features. And so there are many people who've had clinical diagnoses of Rubinstein-Taybi syndrome. Sometimes when I've seen had the pleasure of seeing some of these people in clinic who had those clinical diagnoses, sometimes who were diagnosed by Dr. Rubinstein at our hospital some time ago, we are able to then offer updated genetic testing that they can have to try to find their exact genetic change that's causing Rubinstein-Taybi syndrome. And then people might have had a variety of different tests for those who have had genetic testing. So we'll talk about a couple of these today. So one of the tests that someone may have is called chromosome microarray. And this is a test that looks along our DNA and looks at big chunk. It looks at how our DNA is arranged, which is in general in these big pieces called chromosomes, which is what this is showing here, a big chromosome. And this chromosome microarray will look along our chromosomes and look for any pieces where there is a chunk missing or deleted. And so that is one of the ways we could find the genetic cause of someone having Rubinstein-Taybi syndrome, is that they could have a deletion where this piece of the chromosome is deleted, it's missing, and then they're missing a copy of CREB-BP. Usually, and I should say that to back up, that most people have 2 copies of CREB-BP and 2 copies of EP300. If you have a deletion of one of the copies and not the other, that's when you have Rubinstein-Taybi syndrome. So people with Rubinstein-Taybi syndrome, even if you have a deletion of CREB-BP, you still have one more copy of CREB-BP, but just missing that one copy is enough to cause these symptoms. So another way that we can do genetic testing for Rubinstein-Taybi syndrome is a genetic sequencing test. And so we talked at the beginning about these bases in the DNA codes, C, G, A, and T. And so we could order a test that looks at these bases in CREB-BP and EP300 and look for a change in them. For instance, if you had a deletion of one of the bases that changes the way we're able to read that sequence, or if one of these bases is changed to another, that could result in a protein that doesn't work the way that it's supposed to or doesn't work at all. And then that could cause Rubinstein-Taybi syndrome. So I know you heard me mention Rubinstein-Taybi syndrome types 1 and, oops, sorry. types 1 and type 2. And some of you may or may not know what I mean by type 1 and type 2. So Rubinstein-Taybi syndrome type 1 is caused by having a change in the CREB-BP gene, while Rubinstein-Taybi syndrome type 2 is caused by a change in the EP300 gene. We know that Rubinstein-Taybi syndrome type 1 is relatively more common than type 2, but we are finding more and more people who also have type 2. Okay, so in terms of some of the features of Rubinstein-Taybi syndrome, we know that it's between 1 in 100,000 and 1 in 125,000 people who have Rubinstein-Taybi syndrome. And there are a number of common features, but there also is a lot of variability. And I think probably for those of you who've met other people for support through support groups, or those of you who are able to come to the conference, definitely got to see that just the wide variety in the community that we see in individuals who have Rubinstein-Taybi syndrome. So how someone's impacted in terms of development, in terms of how much developmental delay or developmental disability they have is variable. Intellectual disability is common, but again, variable, and we can't necessarily predict in someone who's very young exactly how they'll be impacted. We just always want to make sure that people have all of the supports and the interventions that they need to maximize their developmental potential and to help them where they need help. But we do know that this is a variable feature. Other common features are not growing as fast and having small size. There are some Rubinstein-Taybi syndrome specific growth charts, which can be really helpful to use, especially if someone is a child and young, and if they're being charted on the CDC growth charts, the WHO growth charts, they're probably going to consistently be smaller, and their pediatrician or their doctor is going to be worried about their growth. And a lot of times when we chart them on the Rubinstein-Taybi growth charts, they're just where they should be. And so that's something that we always strongly recommend when we see patients, is that you be charted on that Rubinstein-Taybi syndrome growth chart. In addition, another common feature is feeding difficulties. We know that some of the characteristic features are large angulated thumbs and broad first toes. But again, this is variable and not everyone has angulated thumbs, for instance. There's a characteristic smile. Some people also have a congenital heart defect. There can be differences with how the genitourinary system formed. There can be differences with how the eyes formed. There also is an increased risk of keloids, which is shown here, which is what can happen sometimes with scarring, where you get this really thick scar. There can be immune system defects and also behavior issues. And one thing that hasn't been looked at a lot is differences between Rubinstein-Taybi syndrome type 1 and type 2. This was from a study, this was some data that was from a study in 2016 that looked at a number of people with RTS type 1 and RTS type 2. And there were some differences that they saw in these groups in terms of how often some of the features were. So for instance, to kind of summarize, because I know this is tiny font, but to summarize, we know that in Rubinstein-Taybi syndrome type 2, there was an increased rate of prenatal growth restriction. So that means during pregnancy of the baby measuring small, or of something called IUGR. There was also an increased risk of preeclampsia in people who were pregnant with a child with Rubinstein-Taybi syndrome type 2. Usually people don't know that at the time. It's only later that after they've had their child and the child's diagnosed, that we know that this is maybe why they had had preeclampsia. We know that with Rubinstein-Taybi syndrome type 2, there's also an increased risk of having a small head size. And there's also an increased incidence of mild intellectual disability, but we still see a lot of variability as well. With Rubinstein-Taybi syndrome type 1, we see an increased incidence of severe intellectual disability, but again, it's still variable, and there are still people with Rubinstein-Taybi syndrome type 1 all across the board in terms of cognition, that this was just more common. We know that in Rubinstein-Taybi type 1, there's also an increased incidence of seizures compared to type 2 and an increased incidence of autism or autistic-like behaviors. And then I have some more data that I want to share that was from, I know some of you may have participated in some online surveys as part of a research study that we had about behavior in individuals with Rubinstein-Taybi syndrome. And this was some of the data from that study. And so these were surveys that people filled out. And so some people filling out the surveys had Rubinstein-Taybi type 1, some had type 2, some had a clinical diagnosis of RTS and didn't know which of the subtypes they had. And then our yellow was kind of the overall average. So we know that of the people that filled out our surveys, which we are very, very grateful for, and this was published in June of last year and is available online. But we know that some of these, as previously published, that there was high incidence of intellectual disability and developmental disability, also of developmental delay. This is gross motor delay was very common, fine motor delay, and speech delay. This was the incidence of autism. So autism also was very common, around 35% in individuals who overall who have Rubinstein-Taybi syndrome, but similar to the other study, we saw it a little bit more commonly in type 1 than in type 2, but you can still have autism in either type. We also see ADHD in all types of RTS. And that looks like it was about 40% in the people who filled out our surveys. We also had a lot of hypotonia, which is low muscle tone, which is a common feature of RTS, and behavior issues were also very common. In terms of psychiatric diagnoses, one of the ones that was interesting that came about was obsessive compulsive disorder. So people who filled out our surveys filled out information about their known diagnoses, they or their family member did. And then in addition, there were a number of other surveys that were filled out looking at anxiety, looking at tendency for OCD or OCD symptoms, and also information about adaptive behavior. And interestingly, so people had been, in terms of people who were diagnosed with OCD, it was about 25%, but we found that when in the survey data that 82% of people in the cohort had a high enough score that they could be diagnosed potentially with OCD. So I think one of the conclusions here was that obsessive compulsive disorder is a very common concern that may be under-recognized, underdiagnosed, and undertreated in individuals with Rubinstein-Taybi syndrome. And this certainly could be contributing to some of the behavior concerns that can come about. We know that anxiety is also a big concern that is very common. We know that panic attacks and separation anxiety and fear of physical injuries were also things that were scored highly in our individuals who have RTS. So we know, which probably may not be a surprise to some of you, and also of generalized anxiety and over anxious disorder. And then we were able to look at this information and look at our different groups of individuals with RTS to see if we could see any differences between the groups. Okay. So between the groups and some of our analyses, so this was comparing individuals with RTS type 1 and type 2. And so in this graph here, individuals with RTS type 1 are in gray, and the light blue on the right is RTS type 2. And so these were showing some of the data from these surveys. And so we saw that individuals with RTS type 2 actually reported higher social phobia than individuals with type 1. But we also saw that individuals, kind of conversely, individuals with type 1 had lower adaptive behavior composites, lower scores in communication and in daily living skills than compared to type 2. We also see similar differences across some of these other subdomains of related to communication, receptive communication, expressive communication, written communication, personal community links and coping skills, that we see lower scores in individuals who have type 1 than type 2, but also notice on these graphs that we do still see for each of these metrics a lot of variability, people who are scoring all over the place. So every person is still an individual. Another thing that we looked at was challenging behaviors between RTS types 1 and type 2. And we looked at this also related to sex to see if there were any differences as well as between the types. And we did see that females who have RTS type 1 seemed to, which is what's shown here in the gray on the left, seem to experience more hyperactivity and non-compliance with directions than females with type 2 RTS. We had lots of people fill out our surveys, which I know were very long, but of course we still don't capture, everyone with RTS. But some of this was information that we thought could be helpful to families. There was also some data that hyperactivity in individuals with RTS peaked during the school age and then seemed to improve at older ages, which was also helpful information. One of our big take homes of all of this data was about OCD and anxiety, and that it may be undertreated and under-recognized in some of our individuals with RTS. So this is our contact information for our clinic. Some of you may know that we're in a transition time right now in our clinic at Cincinnati Children's. Dr. Simpson, as I mentioned, recently had her first baby and is on maternity leave. She is in this center picture here. And she is actually relocating soon to be closer to her family and is moving to Memphis, Tennessee, where she will also still be seeing patients and doing research. She'll be at Le Bonheur and St. Jude in Memphis. And then with her departure, Dr. Rob Hopkin, whose picture is here on the right, is going to start seeing patients with me in our epigenetic syndromes clinic soon. Some of you might have met Dima, our clinical research coordinator. Dima excitingly got into med school and is now a med school student, but wanted to include her picture because a lot of this data was a lot of hard work that Dima and Dr. Simpson and I worked on, as well as, of course, thanks to all of you who filled out surveys. Okay, so I know we talked about lots of things.